Can you explain what metaphase chromosomes are and why they are important?
Of course. Metaphase chromosomes are highly spiralized, short, and thick structures composed of two chromatids. They are most visible under a microscope during the metaphase stage of mitosis, which makes them ideal for analyzing karyotypes. The chromosomes achieve this appearance due to spiralization during the prophase, after DNA replication in the S phase. To observe these chromosomes, mitosis is typically halted in metaphase using mitotic inhibitors, such as colchicine.
What exactly are chromosomes, and what are their main components?
Chromosomes are the genetic units of a cell, consisting of DNA and proteins, and they reside in the cell nucleus. Each chromosome has specific components. Centromere, a highly repetitive DNA sequence where the primary constriction occurs. Telomeres, repeated TTGG sequences that protect chromosome ends. Shortened telomeres are linked to cellular aging and death. Satellite regions, areas of secondary constriction containing nuclear organizing centers, which provide information for ribosomal RNA, rRNA. Chromosomes also have shorter arms called P-arms and longer arms called Q-arms.
Are there different types of chromosomes based on their centromere position?
Yes, chromosomes can be categorized based on the position of their centromere. Metacentric, centromere is in the middle, resulting in equal arm lengths. Submetacentric, centromere is slightly off-center, creating unequal arm lengths. Acrocentric, centromere is close to one end, producing very short P-arms. Telecentric, centromere is at the end, but humans do not have this type.
How do we classify chromosomes into autosomes and gonosomes?
Autosomes are non-sex chromosomes, and humans have 22 pairs of them. Gonosomes, or sex chromosomes, determine an individual's sex. Humans have two types of sex chromosomes, X and Y. Women are homogametic with two X chromosomes, 46, XX, while men are heterogametic with one X and one Y chromosome, 46, XY.
What is a karyotype, and how is it prepared?
A karyotype represents the complete set of chromosomes in a cell, organized by number, size, and shape. Humans typically have 46 chromosomes arranged into 23 pairs. To prepare a karyotype, we collect white blood cells from a blood sample as they contain nuclei. Halt mitosis in metaphase to make chromosomes most visible. Treat cells with a hypotonic solution to burst them, releasing chromosomes. Stain and arrange chromosomes for analysis.
What conditions might warrant karyotype analysis?
Karyotyping is indicated in several scenarios, such as pregnancies where the mother is over 35 years old, repeated miscarriages or infertility, birth of a child with congenital abnormalities, suspected chromosomal abnormalities, unexplained mental retardation or growth disorders, abnormal development of secondary sexual characteristics.
How do scientists represent chromosomal abnormalities in a karyotype?
Chromosomal abnormalities are noted using a specific format. For example, a normal female karyotype is written as 46, XX. A male with Down syndrome due to an extra chromosome 21 is written as 47, XY plus 21. A female with a Robertsonian translocation between two chromosome 21s is 45, XX, Rob, 21, 21.
What is an ideogram, and how is it different from a karyotype?
An ideogram is a schematic diagram of a karyotype. It's a graphical representation that arranges chromosomes by their relative size and shape, aiding in the visualization of chromosomal features.
What is sex chromatin, and how does it function?
Sex chromatin refers to the condensed form of the X chromosome, also known as a Barr body, found in somatic cells of females. During embryonic development, one X chromosome in females becomes inactivated in a random process called lionization. This inactive X forms the Barr body, which can be seen as a dark spot on the nuclear membrane. Interestingly, this random inactivation can lead to phenotypic mosaics in heterozygous females, as seen in conditions like anhidrotic ectodermal dysplasia.
Could you elaborate on euchromatin and heterochromatin?
Certainly. Euchromatin is the less condensed, active form of chromatin that is accessible for transcription during the interphase of the cell cycle. Heterochromatin, on the other hand, is tightly packed and transcriptionally inactive. There are two types. Constitutive heterochromatin, which remains permanently condensed, often found around centromeres. Facultative heterochromatin, which can transition back to euchromatin, such as the inactivated X chromosome in females.
How are chromosomes classified using different nomenclatures?
There are two primary nomenclature systems for human chromosomes. Denver classification groups chromosomes into 7 categories, AG, based on size and centromere position. Paris classification uses banding techniques, such as G-banding with Giemsa dye, to identify individual chromosomes more precisely.
What is the FISH method, and how is it used?
The FISH, fluorescent in situ hybridization, method involves using fluorescently labeled DNA probes that bind to complementary sequences in target DNA. This technique allows for detailed analysis of karyotypes, identification of structural chromosomal aberrations, gene mapping and localization, detection of diseases like leukemia, and monitoring chemotherapy or bone marrow transplant outcomes.
Doctor, what are chromosomal aberrations, and how are they categorized?
Chromosomal aberrations are deviations from the normal chromosome structure or number. They fall into 2 main categories. Numerical aberrations, any deviation from the normal chromosome number. Structural aberrations, changes in chromosome structure due to breaks and rearrangements.
Can you explain the structural aberrations in more detail?
Certainly. Structural chromosomal changes result from breaks and their subsequent rearrangement, referred to as chromosomal mutations. These can be balanced. The total amount of genetic material is preserved, usually without phenotypic effects. Unbalanced. Genetic material is lost or gained, potentially causing severe effects.
What are some common types of structural aberrations?
The key types include deletions. A section of a chromosome is missing, which can be terminal or interstitial. Larger deletions are often lethal. Duplications. A segment of a chromosome is duplicated, leading to extra genetic material. Translocations. A segment is moved to another chromosome, either reciprocal or Robertsonian. For example, the Philadelphia chromosome, a reciprocal translocation, is linked to chronic myeloid leukemia. Inversions. A segment of a chromosome is reversed. If it involves the centromere, it's paracentric. If not, it's paracentric. Ring chromosomes. These form when chromosome ends fuse after deletions. Isochromosomes result from abnormal centromere division, duplicating one arm while losing the other.
Are there any diseases associated with these structural changes?
Yes, several conditions arise from structural aberrations, such as Philadelphia chromosome leads to chronic myeloid leukemia. Chiari-Duchat syndrome, caused by deletion of the P arm of chromosome 5, characterized by intellectual disability and distinct physical features. Wolff-Hirschhorn syndrome, deletion of the P arm of chromosome 4, leading to severe developmental issues.
How about numerical chromosomal changes?
Numerical changes involve an abnormal number of chromosomes. These include polyploidy, having extra chromosome sets, like triploidy, 3N, or tetraploidy, 4N, often incompatible with life. Aneuploidy, an abnormal number of specific chromosomes. Examples include monosomy, loss of one chromosome, e.g., Turner syndrome, 45X. Trisomy, presence of an extra chromosome, e.g., Down syndrome, 47 plus 21.
What causes these numerical changes?
Numerical changes usually result from non-disjunction, where chromosomes fail to separate properly during cell division. This can occur during meiosis or mitosis, leading to gametes or cells with abnormal chromosome numbers.
Thank you, Doctor. This has been very insightful.
Thank you for listening. If our podcast has been valuable to you, we would greatly appreciate it if you could leave us a 5-star rating and write a review on Spotify and Apple Podcasts, or subscribe, like, and share on our YouTube channel. Your support helps us greatly with discoverability. We hope to see you soon.